Hi, Aaron.
Hey, Guoqiang.
Hi. Morning.
How are you?
Good. How are you? Who is not here yet? Okay.
Yeah. Do you want to test out your slides?
Okay, I'll try a little bit. I think there should be no problem.
Hi. Cool. Are you able to share?
I'm trying. Yeah, I'm trying. Let me find the second screen.
Hey, Aaron. Hey, Guoqiang.
Oh,
Hey.
Okay. Do you see my screen?
Yes. Yeah.
Okay.
Yeah. Looks great.
Okay.
Looks great.
Okay. I'll try to find the movie to see how smooth it plays.
Okay.
Okay. Okay, now start the movie. Does it play well on your?
Yeah, I can see it rotating back and forth.
And no jitter. Any jitters or?
Any what?
Any glitches? It's just
It looks
mostly
okay.
looks okay.
Right now I see like another screen. Like I see your other screen on this one. Somehow.
Oh! I moved over. Okay, that's right. Yeah.
Yeah.
Okay.
Yeah, looks good.
Looks okay. Okay.
Yeah, thanks.
Okay, thanks.
Are you in Shenzhen now or Hefei?
I'm in Shenzhen. Yeah, I'm in Shenzhen now. I'm flying to Hefei this afternoon.
Okay.
You're still in Shanghai.
Right now I'm in Yunnan. I'm in Xishangbana. I just went on a little trip there.
Okay.
Who do you want to test your slides,
maybe
please?
want to test.
Yeah, let me check my.
Who has a lot of movies?
Okay, one second. Is it running?
Looks good. Can you do the slideshow?
Okay.
Maybe find the movie.
Yeah, it takes a little bit. This is a big slide and always takes a long time to loading. Hold on, it's coming.
But maybe
Okay,
after. Okay, yeah.
Oh.
Now we see it. Yeah. Good.
Okay, so it's running well.
Yeah, great.
Hey, Guoqiang, too bad I missed you in Shenzhen last week.
Yes, so that's a pity. I was teaching in Hefei, but we found
I
out.
was Guoqiang's lab last week in Shenzhen. It was amazing.
We have something very similar related to whose work. Actually, very complementary.
Hi, Guoqiang.
Hi.
I'm trying to. Hello?
Hello?
You hear me?
Yeah, I hear you.
Hello? Bo?
Maybe your
Oh, it's my,
speaker.
oh.
Your speaker.
Oh. Okay.
Hi, it's long.
I hear you. Yeah.
Okay, okay. Speaker is not connected. All right. Are you at Shenzhen, Guoqiang?
I'm in Shenzhen now. Going to have
Nice.
this afternoon. Yeah.
Nice.
It's warm.
Yeah, actually,
It's actually not so. The last couple of days, it's temperature is dropping.
Okay, great, nice.
It's raining a little bit and it's dropping, but still warmer than Hefei. So I need to take more clothes over. Yeah.
We should visit Shenzhen. Well, you are the
come. Yeah. Definitely. And Aaron, you've been in Shenzhen, but not recently, right?
I was in Shenzhen a couple months ago.
Couple months ago. Oh, okay.
Yeah.
Okay. Yeah.
Have you seen elephants around?
I've seen peacocks and monkeys so far. I mean, Sichuanbana.
So you saw elephants around?
No elephants.
No elephants. Yeah.
Okay. Okay. So my imaginations for Sichuanbana.
Yeah.
Yeah. I actually didn't want to I didn't want to go see I didn't want to go have like my kids ride elephants or things. I'm not really into that, so we didn't see elephants.
Okay.
But the weather is great here.
Yeah, sweet.
It feels like Thailand.
How long have you
It's very close.
a week?
About a week, a little bit more. If we like it, we'll just stay longer. I rented this house on Airbnb, so it's a nice house here, like in the mountains.
Wow, looks really nice.
In China is much cheaper. Usually it's very hot season. It's very busy season usually, but now it's usually it's very hard to rent a house.
There's no one here.
This season.
There's no one here right now. Yeah.
Yeah. We enjoy without the crowd.
Yeah, sweet.
I'm gonna miss your.
I haven't seen you. A long time.
Long. I'll miss your talk on Wednesday.
Sure, no problem. It's, I'm keep giving this week and next week. But we had an annual meeting coming last week, like 3 days straight. Yeah.
Oh, nice.
Very similar talk to again.
I met Guoqiang in Hefei.
Okay.
The cell biology meeting.
Yeah, we have a tons of friends accumulated in Shenzhen recently.
Yes.
You have a young friend, but I have a friend. More and more. Yeah, I haven't been there for a while. But you
come,
very
come anytime.
Sweet.
Next week or
surely we are we just Shanghai without any middle risk region from today.
Oh, just from today. Okay, good.
So, yeah.
So then now you can come definitely.
But still, I've been in Hangzhou last week, so I have another star.
Oh, take the test.
I just have a twice CIBR test to show. Yes.
Oh, no star anymore. Yeah, no stars.
Oh, yes. Cool. No star.
My wife has like 5 stars because she's been to like Hangzhou, Beijing, Guangzhou.
Okay.
Yeah. It'll take more time to clear.
Yeah.
For all the regions. Yeah.
We test everywhere.
Great. Okay, Zhang, should we get started?
Sure. Go ahead.
I'm ready to start. Who will be just doing the first?
Who? You'll be first.
Who?
I'll just say a little introduction. Yeah.
Okay.
Okay, so thanks everyone for joining again. This is Neuro Zoom Yunnan Province edition, and great to see everyone. Before we get started with the talks today, we actually have two great eye candy type talks where the images and movies are going to be really stunning. But just a quick announcement that we don't have any Neuro Zoom next week because of the Christmas holidays, but we have in store for you some really amazing talks in 2022. So Zhang and I will keep bringing you the best neuroscience in the world, keep coming to hear the talks, and more importantly, volunteer to present your work. Just send me or Zhang a note. We'd love to hear your latest. So stay tuned for a great set of NeuroZoom talks coming up. And remember, next week, no talks. Okay, with that, I'd like to introduce our first speaker today, Dr. Hu Zhao. Dr. Zhao is a dentist. He received his undergraduate studies in dental sciences in West China University and also did a dental degree in Sichuan University. Then came to the United States where he did a master's degree in Virginia, and then he did postdoctoral training at University of Southern California. And then he did a second dental degree at UCLA, and then continued research at USC. And here, he really pioneered the discovery of a new type of stem cell associated with teeth in gums that are actually able to give rise to all of the craniofacial bones. And it was a major discovery, and he's been working out the signaling mechanisms involving Gli transcription factors and Wnt signaling. But the reason he's here today is because when I was visiting Beijing, I saw him present this new tissue clearing method. This is a method that perhaps only a dentist could develop because they're interested in embedding and working with bones. A method that, well, he'll tell you about this method. It's spectacular. All of our labs will be using it probably by next year, and it's a way of seeing the spinal cord and sensory nerves in unprecedented detail. So I'm really excited to hear this talk. Go for it. Oh, and just to mention, he was on the faculty at Texas A&M University, and then very recently, a few months ago, moved to Beijing, where he's an associate investigator at the Chinese Institute for Brain Research.
Okay, many thanks, Aaron. This is great. I'm really feel just this is a great opportunity and to share our research with the neuroscience community. Because as Aaron said, I'm a dentist. I didn't have too many connections with the neuroscience field. So I really appreciate this chance. So today I would like to share with us the tissue clearing method we developed in our lab, the TESOS method, and the application of this method on the neural connector mapping. Specifically, we use this method for mapping the individual sensory neural axons from in the adult mouse from digits to the spinal cord. So a little bit of background information about tissue clearing method. Tissue clearing has been in the field for many years, and there have many different methods being developed, and each method has its own advantage and disadvantages. So my lab, when I was in Texas A&M, I was in dental school, so I was thinking about introducing the new neuroscience technique into the dental field. So I tested a lot of methods and I was not very happy with them. And then we decided to develop our own tissue clearing technique. So that was the PEXOS method, which is very traditional tissue clearing method. Basically, just immerse the sample, hard tissue sample or soft tissue sample in different chemicals step by step, and finally reach pretty good tissue transparency for both bone, teeth, or brain, and all kinds of soft tissue organs. So this is already published, and it was pretty popular actually. But as you can see from literature that all the tissue clearing method initially they were developed in order for neural connect mapping. That was the first goal for tissue clearing method. But unfortunately, many years have passed, and you can see that none of the tissue clearing method were really able to perform this task, because there's a fundamental limitation for all the tissue clearing strategies. Because no matter how transparent your sample is, you inevitably lost resolution when you go deep. So you can see in many publications that they claim, well, they can see like one axon at like 7 or 8 millimeter depth within the brain, but none of them can claim that they can see every axon. So that means they can see one individual axon, specific axon, but they can't guarantee to see every axon. So that's why that's why this tissue clearing method really just can't be just doesn't all tissue clearing method don't have the confidence to detect the projection course of axon within the whole brain. And this is one image we did. So we performed the signal noise ratio for in the brain sample. We keep laser intensity the same, and you can see that at the surface region you have very good transparent signal noise ratio. We use 2 different methods, PEX and UDisco. And when you go to 1.5 millimeter, the SNR drop very significantly. And when you go deeper, and SNR just keep dropping. Of course, you can just raise the laser intensity to raise the signal, but this is you still lost a lot of signal inevitably. Some of the detail cannot be seen. Another limitation is from the objective. As we know that the good objective like 40x 1.3 and they have very good resolution, but they also have very short working distance. This is a big contradiction. An alternative solution is like you can just customize made very expensive long working distance high NA objective, but this is not always the case for always a good solution. So currently, actually, there are very limited solutions for connect mapping, and they are all based on a section strategy. FMOS is the one developed by Qingming Luo's lab, and basically it's just a section reconstruction strategy. And GNU4 mouse light project is a combination of a partial tissue clearing with sectioning strategy, and it's a block face imaging. It's not a section reconstruction. And Dr. Guoqiang Bi's lab, they recently published the paper. It's also based on a block surface imaging strategy. So I think we all realize that in order to achieve whole brain equal resolution throughout the whole brain, it's inevitable that we have to section the brain. We can't guarantee just image and intact whole brain sample without any section and keep the same resolution. Is impossible. So when we started our test of this new tissue clearing project, we were aiming at some more unique goal. So as you see that there have been several strategy for brain connect mapping, but there have been none strategy solutions for peripheral nerve. As we know that the peripheral nerve, the sensory terminal can be at the fingertip, and then the axon can travel along many different types of tissues, skin, hair follicle, muscle, fat, bone, and enter the spinal cord, the dorsal root ganglia, and then spinal cord. It's a very complex tissue type, and no strategy actually currently is available for peripheral nerve mapping in the rodent model. So we developed our TESOS method. In brief, TESOS method is the combination of tissue clearing and block face imaging strategy. So I keep saying the complete protocol and recipe is already available on BioCat. Just search TESOS and you will find out. So we're just really happy to share this method even before it's officially published. So in this TESOS method, we propose a new concept, transparent embedding. So the first couple of steps of our TESOS method is pretty much the same as traditional tissue clearing. You do the fixation, decolorization, deliberation, dehydration, and the final clearing medium. The clearing medium has a very special formula. It's composed of benzoate, BB. BD is a bisphenol A, diacrylate. It's a dental monomer. As Aaron said, I'm a dentist, so that's the reason I could think of this monomer. It's a monomer and then plus some UV initiator. So this solution in its liquid form, it can render the tissue highly transparent. As you see here, the brain before clearing, after clearing, in a solution is very transparent. And all the other samples, mandible, vertebral, all kinds of samples, you can read it's pretty good, clear medium. And the unique thing is that then we use UV light to irradiate the sample for like 10 minutes. And UV light will initiate the radical polymerization reaction and just the cross-link reaction. So the monomer will be cross-linked into a piece of gel. And the cool things here is that after this polymerization, both the gel and the sample stays transparent. So that's what we call transparent embedding. And this kind of a formula is actually pretty unique. We have tested hundreds of different formulas, and only very few can achieve this kind of transparent embedding. And this can be applied to both brain and heart, many other heart tissue like mandible, before polymerization, after polymerization, and the pore, including the hair, skin, bone, everything, vertebral bone, the bone, spinal cord, after polymerization. So after polymerization, the strength of the sample, specific here, the brain, could be improved by nearly 200-fold. So the 200-fold means it's actually even stronger than a paraffin wax. If you have experience with the paraffin wax embedding or section, you know the wax, how strong the wax is. So this kind of strategy can be also applied to all kinds of other tissues, soft tissue and hard tissue, or even the whole body. And we carefully design our formula so that the endogenous GFP fluorescence can be very well preserved after the clearing and the polymerization process. Then we also design all these equipments because when we designed this equipment, we didn't have too much resource. So we try to make every equipment simple and straightforward and can be affordable by a regular lab. Like here, we just use door lab magnetic ring mount, put this ring mount base under an upright confocal microscope and image the top part, and then transfer this magnetic mount onto the microtome, and use microtome to section the top part and move back to the microscope. Another strategy is using the milling platform. Because the gel is really strong, so we can actually use a milling motor. You can just put some on this linear guide, image it, and move it along the linear guide to the motor. Use the milling motor to mill off the surface and move back. So this strategy is aiming for the future automation. And then we also design some software, and this is actually the pipeline and sample embedding and the imaging top part section or the milling off. And so we acquire different stacks, and we use the software to stitch them back in the intact stack and for data analysis. So this is actually a complete pipeline. So I don't have too much time to explain detail, but the key point is that we try to use commercially available software or open software to achieve this goal, because initially we didn't have a resource to write our own code or just develop very big data analysis database. So we just try to make everything simple and doable for a regular lab. But the hardware requirement is still pretty high. You better use the high performance workstation. I think it's pretty much available in all the image core facilities. So here I'm showing the process of mouse brain embedding, transparent embedding and section process. Just show you how a transparent embedded sample looks like. Oh, hold on. Sorry, give me 1 second. So this is brain after clearing and UV light. This is gel. And we put it on microtome and sectioning. 5 micrometer increment, 10 micrometer increment. And as you can see that this the section surface is very well embedded, and the surface is very smooth. So this is the final effect. The impact of this is transparent embedding on the imaging quality. So on the left side, the vertebral spinal cord sample was the image with the 20x BAP lens. Working distance is about 2 millimeter, and the image from top to bottom 1.5 millimeter. And you can clearly see how the signal become dimmer and become just blurring to deep region. And zoom in, look the soma region, you can see the background looks pretty blurry. And the same sample we processed with the Tesla's method and the image with the 40x objective. And we made A section and stitched them back together with the image J. And you can see all the axons are very continuous. We actually leave the stitching line there for us for further analysis. But the structure is very continuous. And look at the same soma here. Compared with the left side, you see a lot of more details. And the soma, the so-called background actually turned out to be the signals with a lot of arbor and boutons. And then we did a lot of imaging. This is a brain sample, a small brain sample, 1 by 1.5 millimeter. Taiwan GAP. We imaged with this Leica SPA. So all the image you will see in my presentation was done by a regular commercial confocal microscope, Leica, Nikon, Olympus, Zeiss. We have tested all the 4 major brands, and also 2 photo microscope. This was done with a 40x of 1.3 NA objective. Resolution is 0.2 by 1 micrometer. So although we made 8 cuts, you can definitely, it's hard to tell where cutting lines are, because the stitching can be really just seamless. So the section process didn't bring in any structure distortion because of the embedding. The transparent embedding is very important for the successful stitching at the end. At 200 micrometer, you can see the bouton and the dendritic spine. So for this image stack, we spent about 8 hours, but now we can definitely spend much less time by optimizing the parameters. And 500 to 700 micrometer depth, pretty much the same resolution and same kinds of details. And this is whole body imaging. It's a Taiwan YAP P5 PUBs. We use 20x objective, including the skin, muscle, bone, everything in place except internal organ. We remove the internal organ because Taiwan line doesn't have a signal in those internal organs. And spinal cord embedded within the vertebral bone and dorsal root ganglion. The blue color is autofluorescence. We use autofluorescence to outline structure and also to enhance the signal. So we made about 20 cuts for this image stack. The sample size is about 3.5 centimeter by 1.8 centimeter. And optic nerve, trigeminal ganglia, spiral ganglia embedded within the bone. And the spinal cord embedded within the bone. We remove the brain because for Taiwan YP16 mice, the brain is just a whole bunch of signal and there's no detail at all. It's very disturbing. DRG embedded within the bone. So with this method, we really don't care about any tissue, bone, teeth, which simply just cut them off without introducing any distortion. And this is spinal cord. This is solid hedgehog CIBR AI140 mice induced with tamoxifen, and these are all endogenous GAP. So we use 40x objective image this part of a cervical vertebra containing 3 pairs of DRGs. And the voxel size is 0.4 by 1.2 micrometer. We made about 20 cuts, and this is a Z dimension. And this sample has all the tissues in place, bone, muscle, fat, nerves. That's why we can see the connection between the DRG and the spinal cord, the intact connection. And the final, the boton and arbor. And then we image the peripheral tissue. So this is a mouse pore, also imaged to 40x objective. And we made 20 cuts, 30 cuts. It contains hair follicle, skin, bone, everything in place. And image with the 40x like a SPA confocal microscope. Bone marrow, you can see all the structures here. This is a see-through in the Z dimension. And this is a micellar corpuscle under the walking pad. And you can clearly see the morphology of the terminal. So CIBR label all the kinds of nerves, but we can distinguish the neuron type simply by morphology because it's 40x objective. And hair follicle, lanceolate ending, innervating the surrounding the hair follicle. And bone marrow. So we want, this is slide two, showing the structure is continuous. And we also perform the analysis because we are not just want our picture to be beautiful, we want to be useful. So we use VAR3D for analysis. Because of high resolution, again, we can actually simply trace every single axon and all their terminals in the space. And here is the complete projection of the sensory neurons within the spinal cord, labeled the AAV. We inject the AAV into the pore and retrograde label the DRG sensory neural soma, and together with the entire projection within the spinal cord. And this is spinal cord in place, containing the vertebra and surrounding muscles and every tissue in place. And soma here, and the central axon and their bifurcation, and rostral projection, the end, and collateral branch, all the way to the arbor. And the caudal projections. So because of the details, we can easily just trace every part of this individual sensory neuron. And finally, we were able to outline the complete projection of the 12 sensory neurons within the spinal cord and to perform alignment with the Allen atlas to see where they are projection into. I believe this is actually the useful information finally we want to achieve from those very long and very intense imaging process. And that's the final challenge. The complete projection from the pore to the spinal cord using CIBR YIP mouse adult. We image this sample with a 40x objective. This is a very long imaging process. We selectively imaging just the some of one nerve branch. And we were able to tell the neural type simply by morphology because it's the 40x objective. Simply look at the end, the lanceolate ending, so we know it's the LTMR innervating hair follicle. And then we trace all of them back into the spinal cord. So the good thing for peripheral neural tracing is that they don't have any more branches in the peripheral organs until they enter the spinal cord. So it's pretty straightforward to trace the axons without considering like they probably gonna turn back, things like that. But still, we lost a lot of axons during our tracing. Finally, we were able to trace 5 sensory neural axons in the complete course. So we are showing the tracing course projection here. The tracing course is about 6.5 centimeter, and each axon is labeled with one color. And this is all their terminus, the sensory perceptive field under the finger, and they project into C6 DRG, and alignment with the central spinal cord projection and complete projection of the these 5 neurons within the spinal cord. So I think this is the first complete projection mapping of the individual sensory neurons all the way in its complete morphology. So this is a very challenging project. Actually, I think one very useful application scenario for a regular lab right now is you can use this technique for, a short course axon projection mapping, like what we just did here. We use like a status 5 confocal microscope to perform this projection imaging for our neighboring lab. And this was done by a first-year PhD student after a very short training. The sample size is about 1/8 of a whole brain. It didn't take too much time, 9 hours. It's not very long, actually. So we use a resonance scanner, piezo stage, and 40x objective, and very high resolution. So to image the projection of some specific hypothalamus nucleus within the midbrain. So actually, this helped our collaborator to find out some new projection map patterns. Actually, what he said is like this completely changed his way of thinking, because previously this kind of images just like show off at the end of the final image. But now I think he's saying that this can be their figure one for them to help them to find out new projection pattern and instruct them for design new experiment, because this is very direct evidence to show there's how one specific nuclear can innovate some other nuclear at the same time, because this is projection mapping. Oh, this, by the way, this is the analysis was done with the VAR3D software. We're collaborating with the Han Chanpeng's group in the Dongnan University. So we are still facing a lot of challenges right now. So the first thing is the imaging speed. We are collaborating with light sheet experts or high-speed 2P microscope experts to improve the image speed. So currently, based on confocal platform and piezo stage, actually it can be very fast already, but still non-comparable compared to the light sheet. So we are trying to combine our Tesla's method with the light sheet microscope. Previously, we have actually, when I was in US, we have collaborated with all the light sheet companies like ASI, 3i, Zeiss, but we finally realized that the configuration for all the commercial light sheet companies is not applicable for Tesla's method. I think Dr. Guoqiang Bi's V-shaped light sheet microscope, that's the right way to go. And we need to make our imaging processes automatically. And the very large data sets is also another challenging for us to go. Again, all the protocol and receipt are available on bioarchive. So welcome to use them, use the method. As I said, I believe this method will be very helpful because it's really easy and straightforward. And you can just do it with your currently available upright confocal microscope and without many major modification. So at the end, I would like to thank my lab members and CIBR for the funding support and great resource. Thank you.
Thanks so much, Hu, for that great new technology and great talk. And we're open for questions. Kai Wan? Or any questions?
I have a question. It's a great talk. I'm wondering if your technology works with rainbow recorders.
That we haven't tested that, but we tested multiple. I think that depends on the fluoropher. At least we know it works for Confetti. Confetti 4 colors. We tested the green, tea tomato, M cherry, ZS green, scarlet, GFP, no problem. And BFP is fine, but we rarely use BFP because the penetration imaging depth is really limited for any blue color. So Confetti, I say it's pretty good. So Rainbow, we haven't tried yet.
Thank you.
other questions?
Yeah, I think people have heard about this talk for too many times.
Okay, so it's very generous of you to share the protocols and all of the recipes online, and looking forward to using this technology. So people can continue to put questions in the chat later if they want, and then perhaps we'll go on to Guoqiang's talk next. Thanks so much, Hu.
Thank you, Aaron.
Cool.
And Zilong. Yeah.
Great. Okay, thanks for great talks. So that has very excited to have another technology talk to aiming to solving the most fine structure. This time with the SNAPs. So it's my great pleasure to introduce my great colleague for over decades, Guoqiang Bi. So Guoqiang has graduated as in physics. So in Peking University, he studied physics in college. After studying physics, he went to US, as well as a bachelor degree in physics again in NYU. After that, he went to Berkeley to study biophysics. So slowly, he transitioned himself to the physicist to biologist by finally end up the moving pools lab in postdoc in UC San Diego for 4 years, where he did the legendary work, where still one of the highest citation where Guoqiang worked with Mumin to study the fine timing issues of the spike timing SPTP, spike timing dependent plasticity. Okay, I'm sure all the people in the field has were aware of. So after postdoc, he started his own lab in Pittsburgh, working with assistant, associate, and full professor. So from 2007, he went back to China and come back to the USTC in Hefei to start his own lab. Come back in Hefei, where he start to use the cutting edge imaging technologies and to study the fine structure. So this year, I believe Guoqiang has amazing work, and probably he would not talk about in this talk, where he used the amazing similar fine structure to study the whole brain, not the transparent, but not structure to study the similar with line sheet scanning of the primary brain, where he studied the fine structure of part of a monkey brain, which is really amazing. But in this talk, he will talk another line of his lab, where he used the cryoEM combining with several imaging technologies to tell us how the fine structure of a synapse is. Okay, so welcome, Guoqiang. Take it away.
Thank you, Zilong. Good morning. First, I'd like to really thank Aaron and Zilong for organizing this fantastic series and for the kind invitation. As Zilong introduced, my lab is interested in synapse and neuroplasticity, and more recently, for the past 10 years or so in China, we have been working on imaging the complex brain structures at two extreme scales. One is on the nanoscopic structure of synaptic molecules, as shown to the left here, and the other is the mesoscopic structure that Zilong just mentioned, also Hu has kind of mentioned in his talk, the structure of the whole brain connectome, and in this case, it's a monkey brain, macaque monkey brain. But today I'm going to talk about the molecular organization inside the synapse as complementary to Hu's beautiful talk on the large scale. And let's see what's going on. So I'd like to start with this slide in many of my talks, and to show the complexity of
Guoqiang, you're muted.
Hi, Guoqiang. Yeah.
Okay. Now it's okay. Let us start with this slide to show the complexity of brain circuitry that everyone knows here in this audience. But the number, I'd like to show the number like 10 to the 11th neurons and 10 to the 14th, 15th synapses. And in contrast, in contrast, one of the most powerful AI model, the GPT-3, has about 10 to the 11th parameters, and they in artificial networks, these parameters are probably equivalent to number of synapses. But even this model, even if this AI model has 10,000 times more this artificial synapses to approach the number of synapses in the human brain, it's probably still far less complex than the real brain, because the biological synapse is much more than a single parameter. In fact, different synapses can not only have different strengths, but or weights, but also can obey different plasticity rules. And this is Zhao mentioned that about this spectrum-dependent plasticity that I studied first with Muming in San Diego a long time ago. And when I observed this data, I just fit them with two simple curves for simplicity. For about 10 years or so since then, I've been mainly working on the computation rules and cellular mechanisms underlying SBT. But there's one thing that keeps bugging me. Although these simple fitted curves can nicely summarize the SBT rule in its simple form, but when we look at the individual synapses that's represented by this individual points here, data points here, there are a lot of variability that we can see. For a given spec timing, the synapse can be strengthened to about 100%, or may not change at all. So this indicates something that these synapses may be different in terms of plasticity. And another example comes from this experiment that Zhongxiao Fu and Xiao Tan in my lab a while ago. We had a collaboration, a very nice collaboration with Pete Cheng of Peking University, and we just try to look at the role of mitochondria in synaptic plasticity. But the main points are reported in this Nature Communications paper. But what I want to show here in this figure is that what you can see is that with chemical stimulation, glycine stimulation, you see some synapses get potentiated. Some synapses are stable, and even further, some synapses get depressed, so some become smaller. So even though they all in these three groups, they all have underlying metal flash. So that's the main point of the paper, that metal flash seems to be able to convert short-term plasticity into long-term plasticity. But in this case, when we did the same chemical stimulation for this synapses in the same dish, they can behave differently. So that indicates that these synapses can be very different. But the reason this heterogeneity is not too surprising, because the structure of synapses is complex, and we all know that, but we just in electrophysiology experiments we try to ignore that, just try to do average. But I realize that this may be important for understanding how why we are so complex. So maybe I really want to see how these structures look like, and how the synapses look like, and how the molecules may organize. The best tool with highest resolution is probably cryo-electron tomography, and this is about 10 years ago, we set up a platform for cryoEM at USTC with the help of two great friends, Peijun Zhang and Hong Zhou. And my students, Chang Lu and Yun Tao, are brave enough to really enter this field, because at that time cryoEM was, or even at least especially cryoET, is not so has not been so popular yet, and it's nobody has been able to get anything done with neuronal synapse. But they spend a lot of time eventually get neurons grown on the EM grade and freeze them, and then use cryoET to take images. And this ET images is similar to CT in the hospital, that you take projections from different angles and get this. Then reconstruct the 3D structure with by a series of algorithms in the computer. Then this is the reconstructed synapse of the previous two theories, and you can see that very fine structures in the synapse can be identified, and from vesicles to microtubules, ERs, and eventually these molecules in the cleft, and also in the postsynaptic membrane. If we look further into this synaptic cleft, we can identify many individual particles with different shapes, and we can also see vesicles that link to the presynaptic membrane that with these filaments. We also see the postsynaptic density. It's not as dense as the conventional EM with heavy metal staining, but still we do see there's a density here that's probably postsynaptic, this PSD and other scaffolding molecules. And when we analyze the shape of those cleft molecule on these molecules on the postsynaptic membrane, we get this cluster that can be averaged into this shape. And this shape is interesting. It's nicely fitted with the crystal structure of AMPA or NMDA receptor. The resolution is not high, so it's high enough to resolve whether this is the AMPA or NMDA receptor, but we do see that the shape fits. So we can, with some confidence, we believe that what we see here, many of those particles in this cluster are probably glutamate receptors. And we also see the structure of GABAergic synapses, and they have a different shape of postsynaptic density that's thinner and more distinct. And then we also see analyze the shapes of the particles on the cleft side of the postsynaptic membrane, and they fall into two distinct clusters. And this red cluster, if we average all these particles, we get this shape. It's shorter and more sturdy, but it fits with the crystal structure of GABA-A receptors very nicely. And we know that in this culture, we don't have much. Glycine receptors also have the same, very similar shape, but we know that in this culture, there are very few glycine receptors, so we think these are mostly GABA-A receptors. We published this in Journal Neuroscience a few years ago, and this results basically tell us that cryoET can really resolve the fine structure of the synapse in their native state, and from the high resolution tomograms, we can identify individual macromolecules, and they probably something like GABA-A receptors can be visually seen based on their shapes. This gives us a way to maybe we can really understand the molecular organization of those synaptic proteins in situ. So to do this, why we choose the GABA receptors for the first try, because there is a quite famous popular model about the organization of GABA receptor and scaffolding molecule, that's Gaffrin, because Gaffrin, based on their interaction among G domains and E domains, people have proposed that they may form a very nice hexagonal lattice underlying the postsynaptic membrane. And if this is the case, we should be able to see very ordered structure of organization of the GABA receptors. So with this in mind, and with the electron tomography, we should be able to address this question, whether how these GABA receptors can organize in the effector synapses. But manually, just visually identify these receptors, it's really tedious. It takes a long time. So Xiaokang Zhang joined us and develop a very very nice algorithm. It's to oversample the subtomograms of the on the postsynaptic membrane, and then we can use classification, just 3D classification of all these tomograms, subtomograms, and get and identify some of those. One of those classes is similar, seem to make sense. Many other classes are just represent noise, but one of these 12 classes shows a distinct shape that's similar to the receptor, to GABA receptors, and we can see that they have five-fold symmetry that's known to the receptors. This is the raw data from the top-down view of the receptor of those particles, and after averaging those subtomograms, we can get a structure of this receptor at about 20 angstroms, and these receptors we can this resolution, 20 angstrom resolution, is pretty low for structural biologists, but this is probably the first in situ structure of those receptors or any receptors. And further, more importantly for us, is that we can use this method to really check the distribution of those receptors on the membrane. So that's what Yuntao has done, is just basically plot out the receptor distribution on all these synapses. And this is one of the examples. We don't really, surprisingly, we don't really see much hint of this hexagonal structure. So then Yuntao spent some time to analyze whether there's any order in this distribution. And the most important thing he analyzed, or simplest thing he examined, is the near neighbor distance. So the nearest neighbor distance between receptors, this distribution show a very distinct peak at 11 nanometers. So that means that these receptors like to be close to its neighbor at a fixed distance, 11 nanometer. So that's some real order. So, and this can be seen also if we choose these near neighbors to average, we do see some very distinct structures. And but interestingly, the distance is fixed. But if we look at the distance between, if we look at the linkage, this kind of near neighbor link between multiple receptors, that one receptor can link two other receptors with the same 11 nanometer distance. But this angle that forms between the three receptors can be variable So it's a more or less a uniform distribution from about 60 degrees to 180 degrees. So that means if there's a link, this linkage is flexible also. It's not the distance is fixed, but the linkage, the hinge is flexible. We can see, we also see this one receptor can link to 2, 3, 4, 5 other receptors, all with the same distance, but flexible hinge. When all these linked receptors are plugged together, we see this organization of different sizes of linked networks of the receptors. And this way we describe this organization as semi-ordered mesophasic organization of GABA receptors. That's mesophase means it's a physics term. It's between perfectly ordered crystal and random liquids. And this organization shows some interesting feature, but to quantify this order inside this semi-ordered structure, we used this Voronoi entropy that basically tests the order between order of this distributed points. And if in a perfectly ordered system, this entropy will be 0, and if in a totally random distribution, the entropy is theoretical entropy is about 1.7 or so. And the real distribution of the real receptors around 1.5, and randomized distribution is about 1.6 or so. This mesophasic distribution, we think it probably reflects some self-organized postsynaptic density. And if this is the case, we should expect that this mesophase density may form some phase separation between the cytosol and or even on the 2D membrane. And indeed, when we analyze the distribution of those receptors, we do see there is a sharp drop there's a sharp boundary only for linked structures, but not for those unlinked structures that red points here. That means this interaction is a key to the formation of this condensate. And this condensate of receptors corresponds very well with the underlying condensate of scaffolding molecules that underneath the plasma membrane. And to understand this mechanism, we did a simulation, the collaboration with Wen Junxia of Jiangsu University, and just it's this Monte Carlo simulation that we put in the interaction parameters between these components. And after many steps, they do tend to form those these condensates, and the distribution of the receptors can be very similar to the experimentally observed observations. And the interaction between receptor and scaffold can also be seen in this high resolution imaging. And that we can even see correspondence between individual receptors and individual scaffolds here. And further, we want to understand to see whether this condensates or some this distribution, mesophasic distribution of the receptors have any functional meaning. And we look at the distribution in their correlation with plasma, with the presynaptic vesicles. And those vesicles, when they are close to the membrane, they have two different types. One is contacting to the vesicle, to the plasma membrane. This vesicle membrane is in direct contact to the plasma membrane. The other is tethered, that has this linked molecular filaments that link the the vesicle to the plasma membrane. And we found that these two types of vesicles have different distributions relative to the this mesophase condensate. And in particular, the tethered vesicles seem to be uniformly distributed, but the membrane contacting vesicles has basically most of the membrane contacting vesicles distributed within this phase border, and very few outside this phase. So that or outside this condensate. So that means these receptors tend to be close to the vesicles that tend to contact and probably release directly towards the condensate of the receptors. To summarize, this subtomogram averaging can review the in situ structure of GABA receptors, and with localization analysis, we found this mesophasic organization of those receptors, and this condensate of GABA receptors may have this functional relevance about, for example, more efficient transmission, and they may also be used in certain forms of plasticity. So these are the students who did the work, and thank you very much.
Okay, great. Thanks, Guoqiang, for amazing fine structural snaps. I was open to questions. Okay, here's a question on the chat board. So, Masa, do you want to ask questions yourself? Oh, okay. It's happy holidays. So now Guoqiang is all observing the still the in vitro condition of the individual snaps, right?
This is yes, it's a cultured synapse, cultured neuron. So in vitro relative to the intact brain, but in vivo for the cell. So I guess at this level for the molecular organization, most of the basic features are probably similar to the real synapses. Other people have done that this analysis in synaptosomes, and they found also many similar features. But so we have been trying to coordinate these different ways of studying these molecules.
Yeah. The reason I'm asking, I believe the in vitro snap should be kind of similar with in vivo one, but in vitro culture, it's hard to capture like other like dopaminergic snaps, right? Something hard to catch.
It's great. It's actually that's another way. There is another way to do this. Cryo-electron tomography for the more native circuit structures. You just use high pressure freezing of those slices, and then cut thin slices under cryo condition, and then you can put that under cryoET to study the structure. But it's a little bit more complicated, and we are going to do this after we finish the culture stuff. Yeah.
Great. Another great. Great.
Yeah.
Out. Great. And some more questions. Oh yeah, please. It's one last talk, right? One last talk in 2021.
Okay. Okay. Then happy New Year, happy holidays.
As everybody seems to start enjoy holidays. We'll have a lot of attendance though.
Oh,
yeah,
okay.
A lot of attendance. Yeah, 180. Any more questions? Okay, yes. Fine. Yeah, people just enjoy the one last beautiful 2 talks of the NeuroZoom and 2 talks of 2021. So Aaron, how about we thank all the attendants has come to the 2021 NeuroZoom, and so welcome all of you come back and after New Year, and have a more exciting 2022. Okay.
Great. Thank you.
Okay, thank you, Guoqiang. Thanks, everyone, for great talks.
Thank you, Aaron. Thank you, Long.
Thanks,
Great.
Guoqiang.